now that we understand a little bit about how muscle contraction actually takes place, we got a few more things to look at here. So we were talking about how sarcomere shortening, right? In a muscle cell, we said there's thousands or tens of thousands of those sarcomeres, and they are going to contract, and when all of them contract, the muscle shortens. Now, you may have read in the book, or you hear people talk about how skeletal muscle has this all or none response. And so what that means is if one sarcomere in the muscle contracts, muscle cell contracts, they all contract. And this has to do with that trigger, right? So as that action potential ran across the sarcolemma, when it triggers the release of the calcium from the SR, if calcium enters the cell, right, that muscular contraction is going to take place and all the sarcomeres are shortened. If there is no signal, if no calcium is released from the SR, then none of the sarcomeres will shorten. So when you think of a muscle having this all or none response, it can be a little confusing to think about the fact that, like, so how do you adjust the strength of a muscular contraction? Well, there's actually a few different factors that play a role here. So one of those factors that goes into how much force, how much tension a muscle can actually produce is the length of the muscle when that contraction is being called for, right? So basically what we're seeing on this graph is that there is an optimum amount of overlap between actin and myosin, where when that contraction starts, right, we can cross bridge, we can pull past the different myofilaments, the actin and myosin, and that contraction is really strong. On this end, if you start out and your actin and myosin are already really overlapped, there's like nowhere to go. And so we can see that really at an extreme end, there's enough overlap that you really can't produce any tension with that muscle. On the other end, the same thing happens if there's no overlap. If there's no overlap, right, myosin can't grab onto actin. They're not actually in physical contact. Now this would be very bizarre, right, because normally the titin will help keep enough overlap, right, that we're at least somewhere in here. But if it helps to think about this, this is definitely this optimum. You'll see this in sport a lot, right? If you go to throw a ball, you don't throw it like that, right? You stretch the muscles here in the pectoralis and in the arm, and that stretch brings you from basically like way too much overlap. The stretch brings you into this ideal position to create force. Or you go to kick a soccer ball, right? You don't just straight leg it. You wind up, you probably can't see my leg, but try this, right? Go pretend like you're kicking a soccer ball. What are you gonna do? You're actually gonna lift your back leg. You're gonna put the quadricep muscles in your thigh on stretch so that when you make contact, you can actually produce more tension. Okay. The other thing that makes a difference in the amount of tension produced by a muscle is the frequency of the neural stimulus, right? So in this picture, we see this green arrow. That is the nervous system, the action potential coming down the neuron and saying, hey, muscle, contract. Okay. Notice there's a little bit of what they're calling a latent period or a lag period before the contraction starts. And that's because when the neuron says, hey, contract, we still need to spread that message into the cell and release the calcium, right? Here's where the actual cross-bridging starts, right? And then we can see this is how much tension can be developed from that single stimulus. Now let's put this in a little context. The amount of force produced by a single stimulus is here, even though the maximum tension is way up there. So an individual stimulus hitting a muscle, it causes a twitch, right? So they're saying tension in a twitch. This is really not useful in the body. But if you start increasing the frequency of the stimuli here, what we'll see is even in this particular case, this is called treppe. In treppe, the stimuli are coming after that muscle cell has gone back down into relaxation, right? Notice it returns all the way to zero before the next stimulus comes. But even then, we can see that the maximum amount of force is increasing somewhat, a little bit. It becomes much more obvious when we talk about wave summation. These stimuli are coming more rapidly, more frequently, and they're coming before that cell has a chance to completely relax. Same thing here in what's called incomplete tetanus. I know this is a little hard to see, but these are more frequent, right, faster than what's going on up there, and so we start getting an even higher amount of tension produced. Most of this seems to have to do with the amount of calcium in the cell. If you let that cell come all the way back down to relaxation, some of that calcium is being brought into the SR, but if you hit it with enough stimuli, you might keep a little bit of it. When it becomes more frequent and you're not letting the cell completely relax, some of that calcium is staying in the cell, and that's probably responsible for this increase production of force. Now, in this case, these stimuli are coming so rapidly that we can produce our maximum tension in this muscle, but it's also like this muscular contraction is fused. So really, like a twitch isn't that helpful, trepe not really, and complete tetanus isn't that helpful. We're probably, most of our muscle contractions are somewhere in this, probably mostly in this incomplete tetanus, right, where we can produce a good amount of force. Now, don't let that term tetanus confuse you. There is a disease, right, that whole step on a rusty nail, you get tetanus, but that is named after this phenomenon. So in tetanus, the disease, this bacterial toxin is making it so that your muscles, they're contracted and they're stuck in that position, right? So it's named after that kind of fusion of the muscular contraction. That is pathophysiologic as opposed to useful in contraction. Okay, so we had the length of the muscle can change the amount of tension, the frequency of the stimuli can change the amount of tension a muscle produces, and then when we kind of zoom out and we look at the muscle as a whole, right, think about this. Like if I want to lift this pointer, right, do I need every single cell in my bicep brachii to contract on this? No, this is easy for me, right? Where if I want to pick up a desk or if I want to pick up a heavy bag or something like that, I might need to produce more force. Motor unit recruitment is one of the other ways that we can see this happen. So that's not the best color. Motor unit. A motor unit is going to be one neuron, right? This one nerve. So they have one neuron that's in purple, one that's in blue, one that's in red, and all the muscle cells that are innervated by it. Okay, and so this is cute. They've color-coded this for you. So we can see that there is a purple motor unit here where this neuron is reaching this cell, this cell, this cell, this cell, right? Red, blue, etc. So what happens is when I want to go pick up something really light, I may only need one neuron to tell a set of cells to pick this up. But if I want to lift a 100-pound weight off the floor, I am going to need all the motor units I have, right, to get involved here. And so they call that motor unit recruitment, that we don't use all of them all of the time, just when necessary. And it's really, to me, it's fascinating that some muscles, the motor units are really small. So for things where you need more precise control, maybe in your hands, the example your book gives is in the eye. A neuron is only in charge of, I think they said 4 to 6 skeletal muscle cells to direct eye movements. That's a really small motor unit. Okay, so that gives you really fine control over where your eye is moving at any one time. The quadricep muscle, that big group of muscles on the front of the anterior surface of the femur, right? A motor unit might have thousands of muscle cells that a single neuron is controlling, right? So kind of interesting that the body is set up that way. It's pretty efficient. Okay. Now, motor unit recruitment can be used to help you pick up items. It can also be used, we do this kind of rotation of motor units. So in a muscle, this example, think about something like a postural muscle, maybe the soleus in your calf, right? You need about the same amount of tension being produced so that you don't suddenly collapse, right? This muscle is contracting to hold me upright. But if you relied on one set of cells to be contracted all the time, they're going to wear out. And so what happens here is what they're showing is, okay, this motor unit did its contraction, and then it gets to go through relaxation, right? Coming all the way down to no tension before going back into contraction. And we're actually kind of cycling through the different motor units so that each gets a very slight, but a slight break. Right? So again, kind of neat there. Okay. Other things that we want to think about as far as muscle physiology goes. When we talk about how a muscle contracts, we have different terminology depending on how this muscle is actually producing its force. So here we're looking at concentric, eccentric, and isometric contractions. So these two contractions are both isotonic. And then this final one is isometric. And so in this case, you're lifting the same amount of weight, but the length is going to change. In this one, the length is going to stay the same. Okay, so here we have a concentric contraction, which refers to, ooh, I just combined that, concentric. A concentric contraction, the muscle is shortening while it's producing the force. This is really common, right? If I pick up this marker, this muscle has to shorten while it produces that force. Eccentric contractions, the muscle has to produce that force while lengthening, right? So as I let this pointer down, right? You don't want it just to like suddenly just like collapse, right? It is controlling that descent. That is called an eccentric contraction. Now in the weight room, you'd see this as, okay, someone doing bicep curls. A concentric contraction brings the weight up, an eccentric contraction brings the weight down. And people who get super into weightlifting, you know, they think one's better than the other. You might see someone in the gym with a buddy maybe helping do the concentric contraction, right? And then the lift is actually that eccentric contraction as the weight is lowered. This last group, the isometric contractions, the length of the muscle doesn't change while it's exerting force. So there was, I think it was like in the 70s, this was popular for strength training, right? Like just like hold the weight in this position, right? Or something like that. You don't see that a whole lot anymore. But this type of contraction is super important in everyday life. This is what your postural muscles are doing, right? So maybe if you stand up, feel on your back, you'll feel these bands of muscle on either side. They're not necessarily going through these big length changes, but they need to be able to hold you in position, right? Exerting that force. Okay. Let's talk a little bit about how muscle is actually going through metabolism. So, and we can look at this in a couple different ways, right? Basically, what they're showing you here is resting muscle, moderate activity, and peak activity. And so we're going to go through this and look at what's different, but it is kind of nice they've put them together in figure 1020 so you can make these comparisons. I'm going to make it bigger so that we can actually see this. In a resting muscle fiber, a couple things that I'd like you to note here, right, is that at rest, of course, we do not need as much ATP. Hopefully we're all good with ATP being this cellular currency of energy. In a resting muscle, when this ATP is being produced, ATP is really not meant for like long-term storage. So we're gonna figure out this creative way to go ahead and store this extra energy in the form of this phosphate group. And so in resting muscle, what's actually really common is to take ATP and creatine, right, and actually move this phosphate over, right? So when we take it off, we go back to ADP, right, triphosphate, diphosphate, and what's called creatine phosphate. Okay? Later, when we see muscles actually trying to do something, we can reverse this reaction, pull that phosphate off, re-energize the ATP, and use it in the cell. But in a resting muscle, right, we might be kind of building up this creatine phosphate system. Other things to notice here, right, is that at rest, skeletal muscle is quite happy to use fatty acids from the blood supply as their fuel, right? Any glucose then that's brought into the cell can actually be stored for later in that form of glycogen, right? Because the fatty acids are producing more than enough ATP, we'll go ahead, put those glucose molecules together in the form of glycogen. Okay, so yeah, I think that's good. That's what's going on as you're sitting here watching this video. Let's see what changes when we start to pick up activity levels, right? So now they're showing what they're calling moderate activity. I don't know, maybe going for a walk, right? Now we see that we're actually using this ATP, right, to run that muscular contraction. Remember, we needed that to bond to myosin in that cross-bridge cycle. So we need to produce ATP. They're not showing the creatine phosphate system on here because really that amount of phosphate lasts like 15 seconds. So what we're gonna see during moderate activity is very much the use of oxygen to do aerobic processes to produce more ATP, right? So we see glycogen being converted to glucose, and then that glucose actually being run through aerobic cellular respiration to make a bunch of ATP. Okay? We also continue to use fatty acids as fuels as much as possible. Okay. The nice thing about this, right, is like humans, probably most animals, can really continue moderate activity for a really long time, right? If you think about the amount of stored triglycerides, even in like a fairly thin person, there is a ton of fuel, like days worth of moderate activity. So even if we run out of that glucose, there's nothing to eat, we run out of the glycogen stores, right, we can run off fatty acids for a long time, as long as we're at moderate activity. When we move up to what they're calling peak activity, oh, I guess I could have just left that circled, right? So this is like running a sprint or something like that. They're saying, man, we need a ton of ATP in order to keep this running. Now they're showing us, right, that creatine phosphate system, but again, this is really only like 15 seconds worth of energy, which probably tells you this peak activity cannot be maintained that long, right? But we'll try to get some ATP formed very quickly out of that system. We also go ahead and start taking ATP out of glycolysis. So if you remember this whole process of cellular respiration that uses oxygen and takes time, but the first step, the conversion of glucose to pyruvate, is anaerobic. It does not require oxygen. And so we can use that anaerobic form of respiration to get quick ATP. Now notice this is only 2 ATP. Down here we're getting 30 ATP. Again, telling you we can't maintain this peak activity for a super long period of time. Okay. The other thing that happens here is as we're kind of short-circuiting aerobic cellular respiration, as we're kind of stealing those ATP early on, what we end up doing with pyruvate is instead of feeding it through the mitochondria, we're actually going to convert it to what's called lactate, or what you're probably more familiar with is lactic acid. Those two are the same thing, just like citrate is the same as citric acid. I mean, if you talk to a chemist, they'll tell you slight differences, but they're interchangeable for us, right? And so what ends up happening is this lactate moves into the bloodstream. It's actually going to get sent to the liver, and then post-exercise, your liver, this lactate goes to the liver, and your liver runs what's called the Cori cycle. And that's where you're actually going to then complete the metabolism. You essentially convert it back to pyruvate. The liver does, you'll get this in A&P too, the liver does a process called gluconeogenesis, where it can actually produce glucose. So in the Cori cycle, essentially we're taking lactate from all these muscles, and we're converting it into glucose. That glucose then goes into the bloodstream and comes right back to the muscles. Okay, okay, so this is kind of cool that muscles have all these different ways that they can act depending on essentially the intensity of the activity that's needed. Okay. The other way you might think of this is if you thought of someone doing going for a run, maybe not like a full-on sprint, but maybe not a marathon, but somewhere in there. As soon as you start that activity, even if it's going to be maybe a more long-term activity, you will use up, there's a little bit of stored ATP, a few seconds worth. The creatine phosphate lasts about 15 seconds. You might see that that person can move into this anaerobic glycolysis. You can really only hold on to that for like 30, 40 seconds, and then you really, just the intensity can't stay that high. You're going to have to move into more aerobic metabolism so that you can keep going, right? So sprinters stay in that peak activity, but really probably anything past a 400 meter, you're going to have to move back into aerobic territory. Because the muscles start to fatigue, and when you think about muscular fatigue, it can actually be a lot of different things. It could be the muscle doesn't have enough ATP to keep going. The buildup of lactic acid can cause these feelings of fatigue where you're like, I just can't keep going. The change in pH associated with that lactic acid. You start to kind of get electrolyte imbalances, right? Maybe too much potassium is hanging out outside of the cell, making it so we can't kind of run through those contractions. But fatigue is actually really hard to measure, particularly in humans. If you want to know, because there's a mental component as well. So if you put someone on a treadmill and you said run till fatigue, right, at some point they call uncle. But are they really tired? A lot of these studies were actually done in rats as a study organism, and to get fatigue, they would put them in a bucket to swim, and that rat will keep swimming until it literally can no longer swim, right? As soon as it stops swimming, it's gonna drown, or the scientist will pluck it out, but it will keep fighting, keep trying until it's done. And so then you can measure things like lactate levels and stuff. I don't think they do that as much anymore. I think we got most of that figured out, and we're trying to be nicer to rats. Okay. Okay. Muscle fiber types. Now, the big picture on muscle fiber types is that in different regions of the body, you actually have muscle that behaves very differently. Some of it is actually much more resistant to fatigue. It uses much more aerobic metabolism. We would call those slow twitch fibers, or red muscle, or dark fibers. Fast fibers, right? are often called white fibers, and these are much more easily fatigued, but they can do kind of these bouts of activity. You'll also see people start kind of associating numbers with them as well. So what they're calling here a slow fiber is a slow oxidative fiber, also called a type 1 muscle fiber. Now, a couple things that we can tell, it is slow twitch, right? Meaning, like the gastroc here, to get a twitch takes more like 20 milliseconds, where our eye muscle that they're showing you here as our fast twitch is only taking 7 or 8, right? So we call the gastroc a slow twitch muscle, but 20 milliseconds, I think is pretty good. It seems mean to talk about that way. Anyway, so these slow oxidative fibers, they have high endurance. They are extremely aerobic, right? Think oxygen. They have lots of mitochondria, right, to kind of keep up with those ATP needs. Lots of mitochondria, a really rich blood supply, right? Capillaries are very abundant in here, which is probably part of why it's considered dark, right? Lots of myoglobin. That also probably makes it a little bit darker muscle to hold on to the oxygen, right? Fast twitch muscle, come over here, are also called type 2 fibers, or fast glycolytic fibers, right? Again, the twitch is much faster, the contraction is much faster, okay? But they are very anaerobic, right? They like to do glycolysis and get that quick ATP. They tend to have a lot more enzymes for that creatine phosphate system. Right? They don't have as many mitochondria, they don't have as rich a blood supply, they don't have as much myoglobin. Okay? There is also kind of an intermediate between here. So, and so some people call this type 2B, and there's like the intermediate, they'll call it type 2A, if you want to go with that. I'm okay if you just want to call these intermediate. And so the thing to keep in mind here is this is really genetically determined, and some individuals do have more slow twitch fibers and some individuals do have more fast twitch fibers. So when you think about like an Olympic level sprinter, honestly, genetically, they were probably endowed with more of these type 2 fast glycolytic fibers. If you look at an Olympic marathon runner, right, they've become this good. They've made the Olympics probably because of that genetic predisposition. But it's also type 1 is kind of set, type 2B, those are kind of set. These intermediate fibers, though, seem to respond more to training, right? So they're kind of that in between. If you do a lot of endurance training, they can kind of shift, right, to support that kind of activity. If you're practicing, to be the hurdler, right, a sprinter, that sort of thing, you can actually shift some of these intermediate fibers to be more fast twitch. So there is a genetic predisposition, but these intermediate fibers can kind of be trained in one direction or another. So that's kind of fun. Also note, all muscle fibers in the same motor unit, so all muscle fibers directed by the same neuron, are going to be the same type. There's not a mixture in there.